Hello, thanks a lot, Cici. Yeah.
Can
Hello.
hello, can I can hear you.
Okay. Okay. Perfect. Yeah. So just want to asking some question before on the audience here can ask the question. Mấy bạn ơi, suy nghĩ câu hỏi đi mấy bạn có thể gõ vào trong chat nếu mà mình không có mở microphone, không có tiện mở microphone lên, còn các bạn có thể hỏi tiếng Việt, xong rồi ở đây có Phương, có bên RI sẽ giúp dịch sang tiếng Anh ha, mọi người cứ thoải mái lên, đừng có ngại ha. Thì chắc là bên chỗ của Research Instrument là distributor ở Việt Nam thì có thể cung cấp thông tin, ờ, email hay là địa chỉ liên lạc á, để mọi người có muốn hỏi thêm về cái platform này á, thì có thể hỏi liên hệ với lại bên RI. Ờ, Không biết là bên RI có thể đánh vào cái thông tin trong cái chatbox được không, để cho mọi người có thể có thông tin để có thể liên lạc được. Rồi, trong lúc mà mình hỏi á, mọi người suy nghĩ câu hỏi đi nha mọi người ha, đừng có ngại. Đó, tại đây là cơ hội để mình hỏi mà. Mình đã mời được người ta tới nói để mình hỏi, để mình hiểu thêm ha. So, my first question, so because we know the nanopore technology from the Oxford Nanopore platform. So, CC, there any different, big difference between the cyclone 6 and nanopore, Oxford nanopore sequencing?
You know, you mean the big difference from between the cyclone-seq sequencing and the nanopore sequencing, right?
Yes, the Oxford nanopore sequencing, the Omnium also have 50 gig for the same capacity. Yeah.
Okay, so 小健那个研发的老师想回答吗？还是我来回答？
他是在问 Cyclone 和 ONT 有什么不同吗？
对，差不多。
那你来回答吧，海外你掌握一
哦
下。
Okay. So I'm trying to answer a question. So,
Mm-hmm.
actually, the nanopore sequencing is a general name,
Yeah.
such as the NGS. Yeah, it's a method of the long read technology. So we also introduced the nanopore sequencing in our CycloneSEQ system, but the core protein, such as the multi protein and also nanopore protein, is self-development.
Yeah, I understand that.
Yes,
I just want you asking that the Oxford nanopore also using the pore to get
yes.
the sequencing, and also your technology also the same. I just want you asking that they any different or they totally the same.
Uh, I, I, I think the basic principle of nanopore sequencing is similar.
Okay,
But
perfect.
yeah, but we have the different compounds and the different maybe the base calling method, something like
Mm-hmm.
that. Yeah,
Okay, okay.
such as because the Illumina or some other company have the similar principle for the sequence NGS sequencing. Yeah, similar like that.
Yeah, yeah. And then my second question concerning about uh, the capacity of the machine. So
Mhm.
in the Omnium, the machine have they said that the maximum uh, capacity will 50 gig generate will be 50 gig of data output, and your
Mhm.
machine the CysloneSEQ also 50 gig. So it's the
Yes, yes.
maximum. Can you tell me the average one?
The average, I think this depends on the different application and
Mm-hmm.
also depends on the DNA quality. Uh, so I think for the cultured bacteria, currently the data from our internal testing and also the customer side, the average output is around 15 to 25 GB per flow cell.
Okay. So it means
Yeah.
like from 30% to 50% of 50 gig.
Currently, maybe, yeah.
Yeah. Okay. So, and then in your case study about the thalassemia, you,
Thalassemia.
yes.
Yeah,
Can you please share the slide again, please?
sure, sure.
So I can see that over there you use the whole genome ultra-long amplification of the target. So just my curiosity that how long we can generate the ultra-long. Ultra-long it means that how long the maximum we can get for the target.
Oh, you mean this one?
Yes, exactly. So you can see that they have the ultra-long of the PCR amplification. Yeah. So
Uh-huh.
for my curiosity that I want to know that how long we can do.
How long we can do?
Yes. Like for example, I want to do some kind of amplification of the
Uh-huh.
um, rare population of some species inside the community sample. Yeah.
Uh-huh.
So and I want to amplify the whole genome of the some bacteria around 2.5 megabyte to 5 megabyte. It's possible to do the whole genome of the bacteria
Uh-huh.
amplification. Can we do it or
Uh,
not? It's possible
Shall,
to doing so.
okay. I think I got your question.
Mhm.
I want to ask the R&D professor to answer this question because it's more detailed.
Okay, okay, okay.
小健，你可以让那个老师帮忙回答一下这个问题吗？
他是问这个地平扩增子有多长吗？
哦，对对对。
这个地平扩增子，张佳老师可能也不知道，因为这是股份的项目。可以这样吧，就是我们到时候收集一下信息，到时候他需要的话我们发给他。这是股份，
如果
这是合作伙
他自己做2.5兆，其实是可以的，对吧？如果其他的他做2.5兆，
单独的2.5兆吗？
对，扩增子的长度到2.5兆。
我们最大的扩增子长度大概就在1兆，然后有比较好的数据的话，大概在600 KB 左右。2.5兆可能需要喷一下，需要拼一下。
拼一下，就是没有办法扩完是吗？
它2.5兆 PCR 也扩不完，它 PCR 扩不了那么长的，是一级。
OK。Sorry, I have talked to our cyclone team because this workflow thalassemia is developed by BGI Research and BGI Genomics. So maybe we can get more details from the BGI Genomics after this meeting and get back to you.
Okay, thank you. Thanks a lot.
Yeah.
And so we also do the WGA for the BGA PGTA um, for the
Oh.
yes support for the IVF, and I really
Uh-huh.
want to know that do anyone that apply the long read for this one,
Oh.
because like
So.
we also want to amplify the WGA whole genome
Oh.
amplification and then do
Yeah.
the long
Yeah,
read sequencing.
how about the length of the applicant for the PGTA? How about the length applicant of your applicant?
Yeah, so some in my case, it's the
Uh-huh.
random, the whole genome of human, whole genome. It's
Oh,
not really
yeah,
amplicon. Yeah.
understand.
Mm.
So how about the length of your applicant in your case?
Um, so I just curious for my the other technology, not for the amplicon.
Okay,
Yeah,
okay,
sorry.
okay.
Okay, so for example, if we do the BRCA1 and BRCA2 instead of thalassemia, they
Uh-huh.
have quite they have like 33 number of exons. and 38 number of exon is quite long. It's very similar to
Oh.
the alpha thalassemia gene or beta thalassemia gene.
Mm.
So and I just want to understand that can we do this technology as well? Very
Oh.
similar story.
Understand. Yeah, I will connect with the BGI genomics and get back to you about this
Okay.
information. Yeah.
Okay, thank you. And then you also mentioned about the DNA methylation also called using the CysloneSEQ technology. So
Uh-huh.
I want to know that how accuracy between the calling from the long read compared to the bisulfite conversion sequencing. They very high correlated or how much correlated it is between um, the CysloneSEQ and the bisulfite whole genome sequencing.
小健，你可以回答一下这个问题吗？
他是说我们在人类基因组促进的时候，N50有多长吗？
好像是
一致
问甲基化是吧？我听这是甲基化是吗？
methylation。
对对，我们甲基化产品没有发布吧，所以我们
对，
直接跟他说，
老师你直
目前还没有
接回答吧。
最终的结果吧。
老师你直接回答吧。
哦，行，那我说也行。
嗯。
Okay, I think I will answer this question. The methylation detection product is still in its research stage, and we have not released the product yet. So, yeah, it's not quite clear what the final accuracy of the product will be, but I hope and I think it will be about 95% of accuracy, like that.
Okay, 95% compared to whole genome bisulfite sequencing.
Yes, but that's only a target, and we are still going after that.
And how long for the read we can do the maximum length of the read we can
The
do
read
for
length will be the same as ordinary whole genome sequencing, like dozens of kilobases.
Okay. So can we do the single cell for methylation, DNA methylation using the CysloneSEQ? Do you think it's possible to do so or not?
Yes, in principle it is possible, but as I said, it's still under development.
Okay. That's it. So I also have my last question about the full length stickiness. So in your result, you saw that the full length stickiness, we only need to do the PCR. We don't need to do the to aggregate on the 16S together to have the long fragment in order to sequencing now. Like the BacBio, they have the technology, they have the kit on Kinet. They can aggregate on the 16S sequence into the long fragment size, and then they do the sequencing in that fragment size. Intercept the quota of the sequencing. Do we have the key to doing so or not?
I think I can answer this question. Yeah, if I'm understanding this correctly, yeah, the packball technology, although I'm not an expert of it, the packball technology first ligates the cDNAs
Yes,
into longer fragments and then
exactly.
sequence
Yeah,
that
to do
fragment.
the full length thickness.
Yeah,
Yeah.
yeah. I think that
So
is because there's an optimum length of their technology of their read. And for our technology, there's no such thing. So we can sequence shorter fragments as efficient as longer fragments.
but
So
the point like you cannot save the pore, isn't it? Because you still sequencing very short one, and then we
yeah,
lose the pore after we sequencing very short one. So that reason that
no,
if we
that's not an
concatenate,
issue. One
okay.
pore can sequence multiple reads one after another. That's not a big issue. So there's no need to ligate first. You just
Okay.
sequence the right cDNA.
Okay, thanks a lot. So I have several questions from the chat box. The first one from Huy Hà. He asking, firstly, thank you for your presentation. He asking that the difference in the time consumption and the yield between the single mode and the consensus mode in the real-time analysis.
Okay. Yeah, for this question, I think, I need to clarify that the difference between the single mode and the consensus mode is not the difference in sequencing. It's a difference in bioinformatics. So for single mode, we analyze single reads, and for consensus mode, it's not like the PacBio consensus sequencing. It's just assembling the reads into a long sequence, like a de novo assembly, and we evaluate the accuracy of that long sequence, that come from multiple rounds of reads. So yeah, the yield is of course, I think it doesn't make much sense to compare the yield because the consensus sequence is like a product made from the raw sequences, right? So yeah, and the time consumption, it takes many hours to do the consensus, depends on your sequencing depth and genome size, and also the bioinformatics tool that you use. So yeah, it's basically comparing the raw signal with the highly processed signal. If I have made this clear.
Okay. So Huy, do you think you satisfied with the answer?
Okay. Thank you for your answer.
Okay. So, and then his second question he asking, because I saw you have the future direction, and you said that you will develop the pipeline and as well as the bioinformatics tool to analyze the CysloneSEQ data. So at the moment, there any available of the bioinformatics tool for it?
Yes, we do. And we are currently developing some common bioinformatics tools for CysloneSEQ, and some of these have been released, including the one of the QC, that is quality control analysis tool called Bamboo, and also we have the variant calling, the human genome variant calling tool, and also we are developing some of the newer tools that are going to release very soon, like the single bacterial assembly tool, and also the small genome de novo assembly tool. Yeah, things like that.
Okay.
Yeah, that's the first things I recall in my memory. Xiaojian and Cong Cong, you can give more examples if I have missed anything.
Okay. So,
And also, yeah, there are also some and many existing tools that can be easily applied to CysloneSEQ data, like the alignment to minimap tool and other tools like variant calling to Clear3. These have been tested on CysloneSEQ data and they yield good results.
okay. And the format file from the CysloneSEQ output is similar to the orthogonal pore output or the format file. Any different file generated from your platform?
The file format
Yeah.
are standard FASTQ files, FASTQ. Yeah, these are generated in the same standard as Oxford Nanopore and the PacBio for compatibility.
How about the raw data?
The
So
raw electronic signals, you mean?
exactly, yeah.
These are not output by default because these consume a very large amount of storage space.
Because like if we keep the raw data in the future, if we develop new algorithm, the AI technology going, so we can recalling the base again for more accuracy. So do you think that it should store it also the raw data or not?
Yeah, in principle, you can do that. Currently, as far as I know, it's not supported yet. And also, in the future, there will be new pores and new motor proteins and the new library preparation kits. So the new algorithms will be optimized for that. So if you have old data, I think it's not very likely that you can apply new algorithms successfully.
Okay. So so we have the other question from Ang. He asking that how many indexes are there in a bad branch?
Currently, we have kits for two types of indexes. One kit for 24 indexes and one kit for 96. Is that right, Xiaojian?
Currently, we already launched the 24 barcode library preparation kit. I think the 96 would be end of this year or
Okay.
early of next year. Yeah.
Yeah, it will be very soon. Yeah.
Thank you. So we have one more question from anh Võ Hoàng. She thank you for your presentation. She concerned about the high risk sequencing combined between a long and short read. So she asking that is doing the sequencing at the same time. What application we should use for?
No, it's not doing the sequence at the same time, but you can do it, of course. It requires two different sequencers, of course, and yeah, you can run it at the same time, and also you can run it sequentially. Depends on you. But the key point is what's the strength of hybrid sequencing. I think it's a good combination, good trade-off between accuracy and length and cost. And for applications like de novo assembly, including metagenomics assembly, I think there's a good balance between long read and short read because long reads generate more continuous assembly and the short reads make the base accuracy higher for the assembly. And if you combine these two, you can achieve relatively good quality in assembly without costing a lot of money.
Okay, so thanks for your answer. So we have one question from Jolina. She asking that if she have a sequence not generated from CysloneSEQ, can she analyze the sequence using your tool too?
Yes, but it's not recommended because there might be a lot of parameters that are optimized for CysloneSEQ2, for CysloneSEQ data. And if you apply these tools on other data, there's no guarantee that you will receive good results.
Yeah, thank you. Jolie,
Yes.
are you happy with the answer or you want to asking further? Please go ahead.
Okay, I have several questions. First, for the cystinase or DNA in the cyclone data analysis, you say that you can go down to the species level. So I want to ask, in which database in the cyclone that you use Do you know the database in this tool?
You mean the database for the 16S Florence analysis, right?
Yes, yes.
那个老师可以回答一下，你知道这个 database？
You mean the specific database used in those slides? Sorry, I'm not exactly sure about this, but we can definitely look up for this and go back to you.
I think the slide is like 26 or something, or more up. Let's see. Yeah, yes, I think you
This
just
one?
passed.
Oh, this one is focusing.
The species, going to you analyze into the species for 16S for.
Yeah, only this one.
Yeah, yes. You just passed the slide. I think.
Oh, you mean this one?
No, not
No,
that.
this is not for the 16S Florence. Maybe we
Yeah.
can check after this meeting.
Yes.
Okay, thank you.
Okay. And can I have the slide of yours? Because a lot of information I want to see, and I may have several questions later in your email. But it's possible.
Sure. Maybe I can send you my email firstly. You can send me your question about this slide. We will have an internal review firstly, and then can send it to you.
Okay, thank you very much. Thank you.
Yeah, I will write my email in the chat box, and you can email me.
Okay.
So, and then thanks a lot. And then we have the question from Hoàng Sơn. Can you please, Sơn, can you please directly asking your question, please?
Yes. So in metagenomic assembly, what's your recommend tool to use for assembly? Like in PacBio, they use the meta MPDG to assembly MAG. So do you have any recommend for this?
You mean the recommend assembly tool for the long range sequencing, right?
Yes.
那个研发的老师可以回答一下吗 ？Hello，
可以重复一下问题吗？我没有理解。
就是组装的那个推荐工具。
推荐工具是指那个红金组组装是吧？
他没有说，你如果有不同的，你就说不同的呗。metagenomics， 对，metagenomics。
Okay, for metagenomics tool, among the existing tools, the current recommendation is to use Kraken for separating the species for binning, and also then use MetaFlyE for assembly. And we have tested this pipeline and it works well. And also unicycler is a good choice.
So for the CysloneSEQ long range, you recommend to use the MetaFly, right?
Yes, that's right.
Yeah. Okay. Thank you.
Mọi người hỏi tiếp nha, còn thời gian nếu mọi người hỏi đi. So next we have thanks a lot for your answer. So we have the other question come from Huy. He asking that are there any guideline available for extraction kit for the CysloneSEQ? where I can find them. It is acceptable to you the kit that is not recommended for you with the CysloneSEQ platform.
Maybe I can ask the first question. We
Mm-hmm.
can have our own extraction kits for DNA, but we have some recommendation our SOP. So maybe you can find it in the SOP. Where can I find them if it's acceptable? Okay.
OK，小健，我大概明白你的意思。Yeah, we currently have our own MGI have our own DNA RNA extraction kit, but we also recommend some third-party extraction kit. Depends on the sample type. We also will involve this information into our library preparation manual. So you can check the menu and get the information for the recommended extraction kit. 对吧？
Yeah, if you use the kit and it's not our recommendation, we can't ensure the DNA quality, so we can't get the enough DNA output. So we also suggest you use the recommended kits for DNA extraction.
Yeah, we recommend you use our recommended extraction kit. If you want to use another kit, please ensure the DNA quality and also the DNA amount can meet the requirement of the CysloneSEQ system library preparation.
Thank you for your answer.
Yes.
Because like we do the whole genome sequencing for some plants, and we fail several times. So that reason we asking for it.
Oh.
Yeah.
Also
Yeah.
for the extraction part, right?
Exactly. And also
understand.
we also don't have the good machine, the good equipment to do the QC for rapid size as well. So that reason that we after the DNA extraction we don't have the QC well for it. So that reason that because also in plant they have some chemical very different from blood or some kind of input material. So that be our concern because we agriculture country, so we do quite a lot of whole genome sequencing for plant for do the de novo assembly first. Yeah.
Yeah, understand.
Yeah. Can you introduce us some slide about the machine, the equipment for do the QC of the DNA after extraction?
Yeah,
Sure.
we also have.
Sure.
Yeah.
Do you recommend some equipment that in order to get the best input data for the sequencing before do the sequencing? Yeah.
你说吧，小健，你说吧。
Yes, we also have a recommendation equipment for DNA quality. Also, and another small equipment. We also have a recommendation for good quality of DNA. So you can find this information in our manual.
Okay, can you please send me our link or something in order to
maybe I can send you after this meeting. Together with the question, maybe you ask in the meeting.
Okay, perfect.
Yeah. Please send me your email address, please.
Okay, I will type now. Yeah, for sure.
Yeah, you can text in the chat box.
Thanks a lot.
Yeah, thank you.
Okay, so so for the future direction, I can see that in the application you show in your talk, mostly for the medical application. So do you, do you really keen to develop everything for the medical, or you also come to agriculture, like plant or environment community that community microbiome? Um, Hello, can you hear?
Hello，
嗯
那个研发的老师可以回答一下。
我没听太懂。
就是那个开发的一些计划，就是我展示了一些 medical 的一些 solution 嘛，就我们是不是真的有做过这些？另外就是对于未来的一些计划，比如说 agriculture environment community，然后之类的，
Yeah, just
就是
want
一些开
to
发计划。对，
yeah, just want to understand that the future direction of the CysloneSEQ
huh.
platform, they want to keen for the medical application, or we extend into the other field like agriculture or environment.
嗯。
Yes, I understand. Yeah. Yeah, currently we are prioritizing the metagenomics. I think this is one of the environmental application fields. And also we are doing other stuff more related to medicine, like the human genetic tests and also the tracing of COVID and other viruses, things like that. Yeah. In general, we are not limiting ourselves to just one or two fields. Yeah, we are trying to find out what our customers need the most, and we focus our development effort to those fields. Yeah, but that takes time. So in time, it will be rolled out one by one.
Yeah, because as you know, like if we really want to apply the CysloneSEQ in our clinical application, we need the machine have the IVD. Yeah, so that is in that my question comes from, like if we want to apply it in the real medical application, we need the IVD for the machine as well as for the reagent as well.
Yes,
Yes,
yes.
we also,
An IVD
yes.
license.
Yeah.
That is very important, and we are spending lots of time in application for one, because we could be the first one to apply for IVD license in the long-read sequencing field.
Yeah, because like we don't want really to want spend a lot of clinical trial on the machine, and then they don't have the IVD, so we cannot do anything after that. Yeah. Okay. Thanks a lot for your information. We don't have any further question from the audience. So mọi người không có ai hỏi nữa hả? Thấy đủ rồi hả? Hay sao, có ai hỏi không? Hỏi tiếng Việt cũng được mọi người, rồi tụi tôi dịch sang tiếng Anh giúp dùm cho, hoặc là hỏi tiếng Trung trực tiếp luôn thì càng tốt. Có ai hỏi nữa không? Rồi không hỏi nữa thì chấm dứt nha, tại vì cũng là mình xin từ 7 giờ đến 8 giờ rưỡi, thì về 8 giờ 32 rồi, đúng không ha? Rồi, again, thanks a lot for the very nice presentation, give a lot of information for us. Yeah. Hopefully that in the future, in the very near future, we can apply the CysloneSEQ in our study as well. As we're really keen to apply in the clinical application. Yeah. Thanks a lot again. So now the time up. So thanks a lot.
Thank you. But I think I didn't receive your email address.
Okay, sorry.
Yeah, please send me.
Okay.
So if anyone have the further question after this meeting, can send the question to my email.
Okay, I just type my email over there. I use the Google email for
receive.
yes,
Okay.
for fast catching up. Yeah.
Okay.
Okay. Thanks a lot again, everyone. Yeah. Have a nice evening. See you.
Okay,
See you.
thank
Thank
you.
you.
Thank you.
Thank you.